Have you ever stopped to think about how you, became you? It all comes down to this incredible microscopic dance that's happening inside your body right this second. Today, we're diving into that process, cell division. It's the very reason we exist.
And it really all boils down to this one question. Seriously, think about it. You started as just one single cell. So how on earth did that one tiny speck turn into the complex person you are today, with a brain and eyes and trillions of other working parts?
While the answer is as simple as it is mind-blowing. It's this process that not only builds us from the ground up, but it's also constantly working to repair and maintain our bodies every single second of every single day.
Okay, so to really get our heads around this cellular engine, we need to start with the basics, the key players and the rules of the game.
First thing to know is that we've got two main types of cells. On one hand, you have your somatic cells. Think of these as your body's workhorses, your skin, your muscles, your bones, basically everything that makes up you. Then you have gametes. These are the special reproductive cells, the ones that carry genetic information to the next generation.
And these two cell types handle their genetic blueprints, their chromosomes, very differently. Your somatic cells are what we call diploid. That means they have two full sets of chromosomes, one from each parent, for a total of 46 in humans. But gametes are haploid because they only carry half that amount, just one set of 23 chromosomes, ready to pair up and make a new person.
Now, a cell can't just decide to divide on a whim. Nope, it has to get ready, and that preparation is called the cell cycle. First, it grows and bulks up in the G1 phase. Then comes the really crucial part, the S phase, where it makes a complete copy of all its DNA. After one last growth spurt to gather energy in G2, it's finally ready for the main event, the M phase, where the actual division, mitosis or meiosis, kicks off.
So let's talk about our first type of division, mitosis. Honestly, the best way to think about this is like a biological photocopier. Its one and only job is to make a perfect, identical duplicate.
And this whole copying process happens in 4 beautifully choreographed stages. First up is prophase, where all the messy DNA coils up super tight into those classic chromosome shapes. Then, in metaphase, they all line up single file right down the middle of the cell. In anaphase, the 2 identical copies of each chromosome get pulled apart to opposite sides. And finally, teleophase, where 2 brand new nuclei form around the separated chromosomes just before the cell officially splits into 2 perfect clones.
So, why is this perfect copying so important? Well, it's literally how we grow from a single egg into a baby. It's how your skin heals itself after you get a cut. It's even how some critters, like a lizard, can regrow a whole tail. It's all about making more identical cells to grow and to repair.
All right, let's totally shift gears and talk about the other type of division, meiosis. Now, if mitosis is a photocopier, then meiosis is like shuffling a deck of cards. Its goal isn't to make a copy at all. It's to create something new, to create variety.
And that's the absolute key takeaway here. The entire reason meiosis exists is to produce those special gametes, sperm and egg cells, that are each genetically unique. This variation is the fuel for evolution, and it's why you and your siblings aren't identical clones of each other, even with the same parents.
The real magic happens right at the beginning, in meiosis 1. During prophase 1, the chromosome you got from your mom and the one you got from your dad actually get together. They hug and they swap little pieces of their DNA. This amazing event is called crossing over, and it's the main source of all that genetic shuffling. The rest of this first division is just about separating these new, freshly mixed up pairs.
After that big shuffle, those 2 new cells go on to divide one more time in meiosis 2. This second part is actually a lot simpler, and it looks a whole lot like mitosis. All it does is pull apart the 2 identical halves of each chromosome, which results in our 4 final cells.
And there you have it. That's the end result. From just one original cell, you get 4 completely unique haploid gametes, each one carrying a brand new combination of genetic traits, all ready for reproduction.
So if you only remember one thing from all of this, please let it be this right here. Mitosis makes identical clones for growth and repair. Meiosis makes unique variables for reproduction. One is all about stability. The other is all about diversity.
And this table just lays out those differences perfectly. You can see the purpose right at the top, growth versus reproduction. And look at the outcome. Mitosis gives you 2 identical diploid cells, while meiosis gives you 4 unique haploid cells.
And it all traces back to that crucial crossing over event, which only happens in meiosis. But these processes are unbelievably complex. And when you've got trillions upon trillions of divisions happening over a lifetime, well, sometimes things go wrong. And when they do, the stakes are incredibly high.
When the intricate control system of the cell cycle fails during mitosis, cells can just start dividing without any breaks. This relentless, out-of-control division is exactly what leads to the formation of tumors, the very hallmark of cancer.
So what causes this kind of breakdown? Well, it can be a whole range of things. Sometimes it's external factors, like radiation from the sun's UV rays or dangerous chemicals in things like tobacco smoke. But other times, the risk can be internal, something that's been passed down in our own genes.
Now, when meiosis messes up, it's a different kind of problem. It doesn't usually cause cancer, but it can create sperm or egg cells that have the wrong number of chromosomes. The genetic count itself is just off.
Remember, that magic number for humans is 46. Every single one of your body's somatic cells should have exactly that many to work the way it's supposed to. The required precision is just astonishing.
But if an error happens during meiosis, and a gamete ends up with an extra chromosome, say an extra copy of chromosome 21, then the baby that develops will have 47 chromosomes instead of 46. That tiny change, just one extra chromosome out of place, results in the genetic condition we know as Down syndrome. It's such a powerful illustration of how critical accuracy is in the stance.
So as we start to wrap this up, what we're really seeing is that cell division isn't just one thing, it's two completely different fundamental blueprints for how life operates, one for making copies and ensuring continuity, and the other for creating variety.
Mitosis is constantly building and fixing our bodies, while meiosis is shuffling the genetic deck for whatever comes next. It's a system of truly breathtaking precision. Which really makes you wonder, doesn't it? As our cells continue this incredible dance, what other secrets about our health, about disease, about who we are still waiting to be discovered in its steps? Thanks for joining me.